Hello and welcome to Nikolay's genetics lessons, and today I have prepared another video about Hardy-Weinberg principles, and today I'm going also to show you how to solve problems using Hardy-Weinberg formula. And as usual, I recommend you to stop video here, read the questions, answers, and choose the correct answer, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. And here is the first problem. Hardy-Weinberg equilibrium assumes that in the population, answer A, no selection is occurring, answer B, no mutation is occurring, C, no migration is occurring, D, mating is random, answer E, the mating population is very large, and F, all the above. And the correct answer would be answer F. All of the above postulates would describe exactly situation when population would be in equilibrium. And what does it mean? It means that all the genotype frequencies would stay the same generation after the generation. And next question, if the frequency of the recessive A allele is 0.1, then if the population is at Hardy-Weinberg equilibrium, the percentage of individuals that are homozygous recessive should be, and imagine that this is a gene pool. So what is a gene pool? Actually this is different alleles that is present in the gene pool. Usually not all the alleles are analyzed at the same time, but only one locus, probably with just 2, 3 or more alleles. So imagine that we have here two alleles in one locus. One would be dominant allele A and another one would be recessive allele A. And of course frequencies of these alleles can be different. For example, dominant allele A can be present in much higher frequencies than recessive allele A, and that means that of course we would have much more variants of the people who is of course diploid and would be homozygous dominant, and some of them would be heterozygous and some of them would be homozygous recessive. But because, as you see here, frequency of the dominant allele A would be much higher than frequency of the recessive allele A, we would see people who would be homozygous dominant for the dominant allele A in much more frequencies than people who would be homozygous recessive for the allele A or would be heterozygous. So as you see, frequency of allele would directly relate to the frequency of different variants of this allele in homozygous state or in heterozygous state. So if we know that the frequency of the recessive allele A is 0.1, so 0.1, we instantly know that frequency of the dominant allele A would be 0.9, because here we have only two variants of the alleles. So how to find the frequency of the homozygous recessive variant? So this is our question. We just have to multiply probability of recessive allele A, that is frequency 0.1, to meet another recessive allele. So what we should do, we have to multiply these probabilities. So 0.1 we have to multiply by 0.1, and our answer would be 0.001. Or if we multiply our answer by 100, we will get our answer in percentage form. So this is going to be 1%, and this is going to be our answer. And here is a Hardy-Weinberg formula. So probability of the homozygous dominant variant would be 0.9 multiplied by 0.9. So this is going to be probability of the capital A and capital A homozygous dominant variant, and in Hardy-Weinberg formula p squared stands for this probability. As for the heterozygous variant, we have two variants. First would be capital A small a, second would be small a capital A. Or we can cross out one variant and put 2 here. So we in our formula would have so p squared plus 2pq, and capital A stands for the p in the formula and small a stands for the q. So plus, and the other variant would be 0.1 multiplied by 0.1 probability of the allele A small and another small allele A. And Hardy-Weinberg formula we have here q squared. So A multiplied by A we also can express as A squared. But in Hardy-Weinberg formula instead of small a we have q. So that's why we have q squared here. And all this would equal to 1. So here we can put f, frequency of all 3 genotypes in a gene pool. And of course here I don't have much place left. We just put probability of the capital A, that is going to be 0.9. We have to multiply by probability of the small a genotype, that is 0.1, and we have to multiply this by 2. Once again, because we know frequency of the small a allele or recessive allele, in order to find probability of the genotype to be A or A squared, we just have to multiply probabilities of this allele by itself. In Hardy-Weinberg formula, this would be equivalent of this part. And once again, do not forget to multiply the answer by 100, because all the calculations in Hardy-Weinberg formula is made on the scale between 0 and 1, and if the question is in percentage form, we have to multiply by 100 in order to make our answer on the scale between 0 and 100%. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.